Good evening, everybody. It is my pleasure to welcome everyone once again for our 26th season of Sea Secret. I'm Rani Avissar, the Dean of the Rosenstiel School. Thank you for joining us tonight for this online session. All lectures planned for this year will continue to be online until further notice. And this season, we have decided to focus on the 4 strategic initiatives that the school is leading for the foreseeable future. We have aligned those initiatives around many issues facing humanities, not only what we currently experience with the pandemics, but other issues that we should not be forgetting about. And one of them is to do with feeding the world and the main problem of feeding the growing population of the planet, which has a direct impact on the amount of proteins that we are pulling from the ocean, and as a result, depleting the ocean from its natural resources. The second one is to do with protecting our marine resources, and we think mostly about the coral reef, and as I just mentioned, the fisheries and many other aspects of it. The third one is to do with saving lives. The devastating seasons that we just experienced with a hurricane reminds us of the importance of being capable of forecasting those terrible storms. But it's also associated with the interaction with the climate, sea rise, and other issues that we are taking into consideration. The last, not least, is unlocking our ocean secrets, and that has to do with the fundamental research that we do that help us progress as for the understanding of our planet, that goes way beyond the understanding of the environment, but also as a branch in medical, marine medical initiative. So these initiatives form the cornerstone for philanthropic support of our school. And tonight's presentations from our Cooperative Institute that we have developed with NOAA, the National Oceanographic and Atmospheric Administration, addresses each one of those initiatives. You will hear more about the vital research taking place to further our knowledge of marine and atmospheric processes, knowledge which serves to inform and develop policies. We are very fortunate to be one of the 16 national NOAA Cooperative Institute that is based here in Miami-Dade County. So CIMAS conducts mission-driven research which addresses hemispheric and locally relevant problems, and that's generating actionable real-time scientific solutions. Scholarships allow us to recruit the best and brightest students to our school, and CIMAS provides an outstanding opportunity for us to transform students into the next generation of scientists. Students benefit greatly by learning and working alongside world-class scientists. If you're interested in learning more about how you can help support our students in our 4 key initiatives, please email me, and we are going to put in the chat my email and the website of the school so that you can obtain any information. We definitely want to hear from you. A special thanks to our Sea Secret sponsors and the many friends who contribute to the Rosenstiel School. And that includes our presenting sponsor, the Bank of America, the Shepard Broad Foundation, Bill Galway, Cheryl Gold, Key Biscayne Community Foundation, Key Biscayne Life Enhancement Fund, John McCann Family Foundation. Thank you all for supporting Sea Secrets. Tonight's Alum Spotlight, I have the pleasure to introduce you to Rosenstiel alumni Erica Rule. Erica earned a Bachelor of Science in Biology at Mary Washington College, and then joined the Rosenstiel community in 1997 to study marine affairs and policy. She graduated in 1999 with a Master's of Arts and began a career at the National Oceanic and Atmospheric Administration here in Miami. Erica played a big role in expanding the local and national visibility of NOAA's Atlantic, Oceanic, and Meteorological Laboratory, as their communication director and was a key figure in their science review strategic planning exercises. In August of 2020, she joined NOAA's Southeast Fisheries Science Center as Chief of Staff for Science Planning and Operation. As a member of their leadership team, Erica coordinates and helps implement the strategic planning process, priority-based resourcing, and standardizes policies and protocols for the center. We are very proud of Erica and are pleased to have her with us tonight. Welcome, Erica.
Thank you, Dean Avasar. It is my pleasure to join you this evening. I am a big fan of the Sea Secrets series. When I was a graduate student in the late 1990s, Dean Otis Brown would tease me that I was attending the lectures just as much for the free snacks as it was for the speaker or topic. And now I get to speak to you here as an alumna. So I'm really excited to be here along with my CIMAS colleagues. You go ahead and share my screen. And there we go. And there you're looking at the Virginia Key campus with both NOAA facilities. The Marine Affairs and Policy program at Rasmus prepared me for and introduced me to a career I never anticipated as a new graduate student. I'll never forget studying coastal law with Dr. Moreno, conducting fieldwork on Key Biscayne beaches for Dr. Lutzon's environmental econ class, and training as a science diver with Rick Gomez. Here I also made lifelong friends through outreach events, classes, the Propeller Club, and of course, over beers at the Wet Lab. And these are friends I'm also happy to call my NOAA colleagues today. Of course, all marine affairs students are required to complete an internship for graduation, and I was encouraged to apply to an internship with the Consortium for Oceanographic Research and Education. It was a unique opportunity arranged by Dean Brown through his leadership with the organization. And so I have Rasmus to thank, as well as Dean Brown, for sending me to Washington, D.C., where I learned about the federal oceanographic community and was also involved in outreach events like the National Ocean Sciences Bowl. That internship and connections I made back here in Miami opened the door to my first job, which was actually at CIMAS, where I was hired to support science outreach at NOAA. That CIMAS job led to a federal position that grew over 20 years to encompass many aspects of science communication, from media relations to congressional affairs, science symposium to research reviews. I always loved the opportunity to connect people with the science of coral reefs, hurricanes, and the world's changing oceans. It's also taken me to some pretty cool places, such as the eye of a hurricane on the NOAA Hurricane Hunter aircraft, the nauseating swells of the Gulf Stream on large research vessels, and to visit the shallow reefs of the Florida Keys National Marine Sanctuary. It was also pretty great to find a job right across the street from Rasmus, allowing me to stay in touch with the Rasmus community. I served several years on the alumni board, where I launched events such as the alumni golf tournament and our memorable crawfish boils, all in support of student scholarship. It also allowed me to watch firsthand as this institution evolved and added new programs, such as the Masters of Professional Science. One of my favorite accomplishments with NOAA was developing a social media presence and a state-of-the-art website that greatly improved our ability to communicate our message to target audiences. I never could have imagined or accomplished our communication efforts without leveraging the partnership NOAA has with the University of Miami through CIMAS. Over the past 8 years, I looked to the Rasmus Masters of Professional Science program to recruit graduate students for a paid annual internship to learn with us and support the growth of communications at the NOAA AOML lab. These students brought their passion and their skills in social media, skills that I did not have at the time, and created the organization's Twitter and Instagram accounts, and completely redesigned the AOML website. These students continue to graduate and become gainfully employed in the field of science communications. And they also taught me how fulfilling it is to be a mentor and to help others succeed. And so now, in my current position with NOAA Fisheries, I get to work with a team that provides the scientific advice and data needed to effectively manage the living marine resources of the Southeast region and Atlantic high seas. From sharks to dolphins and queen conch to sea turtles, it's a new world for me that brings me back to my graduate days at Rasmus. I'm also thrilled to join the Science Center at a time when new leadership is looking to the future and driving a lot of change. To effectively meet our mission and deliver to our customers, we are in the midst of an organizational realignment across our 5 facilities, ranging from Beaufort, North Carolina to Galveston, Texas, to better align to our core functions. We've also mapped out a strategy to ensure we are prioritizing where we spend our time and our limited resources. As we enter an era where challenges will require partnerships and creative solutions, we look forward to working together with our NOAA partners, with University of Miami and CIMAS to achieve these solutions. I look forward to facilitating these partnerships, and I especially look forward to hearing from our CIMAS scientists this evening. Thank you all.
Thank you so much, Erica, and a pleasure to have you again with us this evening. Thanks. It is now my true pleasure also to introduce Ben Kurtman. Ben Kurtman is our director of CIMAS, and for those of you, sorry, I apologize, for those of you that are not familiar with all the acronyms. So CIMAS stands for the Cooperative Institute for Marine Atmospheric Studies. RASMAS stands for the Rosenstiel School of Marine Atmospheric Science, and NOAA, I guess, is much more known, but it's the National Oceanographic and Atmospheric Administration. So Professor Kurtman received the Bachelor of Science in Applied Mathematics from the University of California in San Diego, and he received his PhD in 1992 from the University of Maryland at College Park. Then, for a few years, he worked at a Center for Research, which is known as the Ocean Land Atmosphere Center, and was after that an associate professor at George Mason University. We stole him from George Mason University in 2007 when he joined the Rosenstiel School in the department, or at the time, in the division of atmospheric science. Dr. Kurtman is the recipient of multiple awards, both internally within the university as well as externally, and is very involved internationally. Within the university, he received the prestigious Provost Award for Scholarship Activity, which is given once a year by the provost in the entire university. So it's extremely prestigious to get that. And in fact, not only he is a great scientist, he's also a very appreciated teacher, as he has received from the undergrad community the undergrad teaching awards in 2016, 17, and 18. He was then elected a fellow in 2018 of the American Meteorological Society, which is the main society that hosts our activities. In 2011, he serves as the Associate Dean for the Rosenstiel School for a period of 3 years until he became the Director of CIMAS. He has been recognized also at Stony Brook University by receiving the Robert Sess Distinguished Lecture in recognition of outstanding contribution to atmospheric science, also in 2018. As I have mentioned, Professor Kurtman is very well known internationally. He participates and leads multiple scientific panels that are part of the World Climate Research Program. And I will just also mention that he was a lead author of the Intergovernmental Panel on Climate Change, well known also by its acronym IPCC. Professor Kurtman contributes a large amount of his time to the community as well by being an editor of various journals, including being the executive editor of Climate Dynamics. He is currently an associate editor of the American Geophysical Union Journal of Geophysical Research, and he is an editor of the American Meteorological Society Journal Weather and Forecasting. He has received research grants from pretty much every agencies that support science. That includes the National Science Foundation, the Department of Energy, NOAA, of course, NASA, the Office of Naval Research, and so on. He has published at least at the last time that we counted, 220 peer-reviewed papers and is extremely well cited by the scientific community, emphasizing the quality of his work. I could continue for another half an hour to speak about the achievements of Ben, but that would not leave time for the presentation, so I'm going to stop here, and Ben, all yours.
Thanks. Thank you for that extremely generous and embarrassing introduction, Ronnie. Thank you very much. I'm going to be extremely brief. I just want to let everybody know we had this new Cooperative Institute. It was a full-up competition, and we won the award, and we had a public celebration on November 17th, 2020, to celebrate the new Cooperative Institute that's entitled Innovative Science, Service, and Stewardship. The problem with that public series is we didn't really get the chance to show our innovation, our innovative contribution. And so today, that's the purpose of Sea Secrets today, is to show you some of the scientific innovation, the research that really led to the award of the Cooperative Institute. And so there's several CIMAS researchers that are going to make presentations, and I really thank them for their time and their willingness to do this. And I'm sure you're going to enjoy it. Along with the new award, we also had the opportunity to expand the leadership of the Cooperative Institute. And so we've asked Emily Becker, Dr. Becker, to become an associate director of the Cooperative Institute. So we stole her away from a different part of NOAA. She has an extremely long history of working with NOAA, primarily on prediction problems on subseasonal to seasonal timescales. And she has written a number of papers that are extremely well received by the research community and continue to attract a lot of attention. Emily has also an extensive experience in science communication, and that's something that in coordination with Erica's efforts, we really seek to continue to expand. With that, I'm going to turn over the run of show and let Emily do all the introductions as a way of introducing you to our new additional associate director. So Emily, I turn it over to you.
Thank you very much, Ben. That was also somewhat embarrassing, so I thank you. At the risk of echoing all of our previous introductions here, I'm very excited about the talks tonight and showcasing some of the really innovative and interesting work that is done at CIMAS. Our first speaker tonight is Lisa Bucci. She's a hurricane research scientist at NOAA's AOML, Atlantic Oceanographic and Meteorological Laboratory. She holds a bachelor's from the University of Michigan, a master's from Florida State University, and a PhD from the University of Miami, another alum. Her research takes her on NOAA's Hurricane Hunter aircraft, where she evaluates new observing technologies and tests flight patterns. Outside of hurricane season, she integrates new observations in weather models to help improve tropical cyclone forecasts. She was a CIMAS scientist for 7 years before becoming a federal employee in 2019. So Lisa, if you would share your screen, we will hear from you now.
Hi, everyone, and thank you for that introduction. So I'm here to talk to you about the science of flying into hurricanes, and I got this dream job actually through CIMAS. So thank you to CIMAS for both allowing me to advance my academic career and pursue a PhD, but also launching my career as a federal scientist, still doing the job that I love, which is a hurricane scientist flying into hurricanes. So every year, NOAA flies aircraft in and around hurricanes. We have two types: the jet that flies high and around hurricanes and gives us information about where the hurricane is going and how it's being steered, and the P3s that fly low and slow into the hurricanes, and that's the one that I typically fly on. So every year we conduct this intensity forecasting experiment that has three goals. We want to be able to make forecasts better. We're collecting observations for our forecast models. We want to be able to get the most complete picture of what the storm is doing at that moment when we're flying into it. And the best way to do it, the best way to get that information is to take an aircraft into it. So you can't get this information anywhere else. And we call that nowcasting. And finally, we're trying to answer questions and gain a fundamental understanding of some things that we don't understand about hurricanes, like why a hurricane might go from a tropical storm to a major hurricane and rapidly intensify. So the way that we do that is by equipping our aircraft with lots of different information. We have probes attached to the aircraft that are measuring temperature and winds. We have things called expendables, drop zones, that we're actually launching out of the bottom of a plane. And as they parachute to the surface, they're sending back information about the temperature and the winds and the pressure. And finally, we have lots of different radars, including one in the tail that gives us information about what the winds are doing in a hurricane where it's raining. So it kind of gives the hurricane like a CAT scan. So when we take this information, we're sending it to lots of different customers. For the nowcast, the people that want to know what's happening right then in the hurricane, we're sending it to the forecasters at the National Hurricane Center. We're also sending the information to our modeling centers. So the models, the forecast models that you're seeing on the evening news need to know where to begin. So we're sending that information, and those observations are being used in those forecast models. And finally, we're using the information to do research. And we have lots of different partners that we're doing research with. We're obviously partnering with academia like the University of Miami, the Cooperative Institute, internationally, other government agencies. The data is publicly available. So stepping through each of the goals, I'm going to give a few examples. And one of them comes from my PhD research, which not only are we trying to get the most accurate picture with the standard equipment that we fly, but we're also trying new instruments. And one of them was called a Doppler wind lidar, which is actually a laser that measures winds. And it measures winds in low or no precipitation areas. So think of it as a nice complement to that radar, which is giving you winds where it's raining. So we didn't know if this instrument would work, but we tried it out, and it turns out it collected great winds. We were able to validate them and get some nice pictures. And what we found was that the LiDAR was giving us information in areas that we weren't able to measure very well before. Often in the lowest levels of a hurricane, like from the sea surface up to about 1 kilometer, called the boundary layer. We were also getting more information about the winds in the formation stage, in the early stages of hurricanes. And you'd be surprised at how much clear air is actually in a major hurricane, but we were able to collect information in some of the stronger storms between like eye walls and rain bands. And that's one of the examples that I have in this picture here, where we were flying from point A to point B along this rain band that was forming. And what we were finding was the winds coming off Honduras were weaker. You can see that in the blue. And as we started to get closer and closer to the coast of Belize and Mexico, those winds were getting stronger and stronger, as indicated by the red colors. And actually with this flight, it went from a tropical storm to Hurricane Earl in 2016. So it was giving us this great real-time picture. Next, we look at the forecast. And not only are we feeding information into the forecast models, we're also using our observations and comparing them to the models to make sure that the forecast models are representing the hurricane as accurately as possible. So on the left side, where we have the observed, we're looking again at that lowest level, the boundary layer of a hurricane, and we're looking at the winds there. So we took all of our observations and we said, this is what a hurricane boundary layer should look like. Then we compared it to the model, the picture in the center, and we found that the model didn't have a very accurate representation of what the boundary layer was supposed to look like. So we started to dig into all the code and found that we weren't giving the model the right numbers to create an accurate representation of that boundary layer structure. So we went in, we took our observations, we corrected the numbers, and then made a much better looking boundary layer after comparing it to our observations. So not only did we make a better structure in that model then gave us better forecasts. So we're improving our models and getting better forecasts using these observations. Finally, I work at the Hurricane Research Division, and so most of our publications go into the research. We're trying to answer the questions of why the storm forms where it forms, why is it rapidly intensifying. When we broke down all the publications that we were doing, we found that we were often publishing about the structure, the precipitation, the rain, the boundary layer, and we weren't doing as much on some of the other topics, like the turbulence in a storm or the microphysics, all the little rain particles and ice particles. So now there's a push to start filling in and expanding our research portfolio into those areas. So we actually, this year in 2020, we started using the plane and flying in it a little bit differently so that our probes could measure things like the rain and ice in a storm so that we're able to start publishing and answering questions about those topics. So I get this asked a lot, what's it like to fly into a hurricane? This is from Hurricane Lane in the North Pacific as it was approaching Hawaii. It was a Category 5. For this one, it sort of felt like a roller coaster. It was bumpy. Right now, you're seeing what it's like in the eye wall of Lane. And then this was one of the prettiest storms that I ever experienced, where you get into the eye, all of a sudden, you just see a sunny day, blue skies above, swirling crowds around you. And actually, you can even see the ocean crashing beneath you, which is pretty rare. You don't often see something like that. But every hurricane is different. You might have a bumpy ride. It might feel like you're driving down a dirt road. Sometimes it feels like a roller coaster, and sometimes it's totally smooth. So you never know what you're gonna get, but every time I go, you learn something new. So it's my dream job. I love it. So thank you for giving me an opportunity to talk about the research, and I'll turn it back over to Emily.
Wow, thank you. That was fascinating. That video was incredible. Really appreciate it. We will have time for questions after all the speakers have concluded, so please put any questions you may have in the Q&A box, and also please indicate for which speaker the question is addressed. And our next speaker is Marlos Goiz. Marlos is an associate scientist at CIMAS. He did his master's and PhD at the University of Sao Paulo, Brazil. After that, he came to the US on a POGO scholarship at the University of Maryland, followed by a postdoc in the Department of Geosciences at Penn State University. He joined CIMAS in 2010. His research and this talk focuses on large-scale ocean circulation and climate, and he also focuses on the assessment and design of ocean observing systems. Okay, Marlos, you can share your screen, please.
Thank you, Emily, for the introduction. Let's see if I find it. Can you see the slide well?
Yes, we can see them. You're a little quiet.
Okay, I'll try to speak louder. Thank you.
Okay, thank you.
So today, thank you for the invitation. First of all, and I'm really happy to be here and talk a little bit about my work. So today I will be talking about some work I've been involved and some recent results have on the Atlantic meridional returning and how we use observations to improve our knowledge and expand our knowledge of the past variability of the circulation. First, I want to introduce what the Atlantic meridional returning is. So this is we call it AMOC, and it's a global circulation. It's similar to a conveyor belt, and it's driven primarily by temperature and salinity changes in the ocean. And in the Atlantic, it flows meridionally and can be confounded with the Gulf Stream northward, and it carries a huge amount of heat northward and is responsible for modulating the climate, weather, sea level, not only in the North Atlantic, but globally. And future scenarios of the meridional, the strength of the circulation from IPCC climate models, right, numerical models that run the simulation. And it showed that with addition of greenhouse gases, especially in the high emission scenarios, the circulation may drop by 60% of what is today by the end of 2100. So it can have dire consequence for our climate. So what we see nowadays from our long records, we can see from sea surface temperature is one of our long record, instrumental records. And we see here if you look at the trends since 1900, we see this picture, like the globe is mostly red, it's warming, and it's some of the regions up to 2 degrees Celsius per century. And there's a little feature in the North and North Atlantic close to Greenland that is actually cooling. And people, some works have linked that to this strength of the circulation. And for this pattern, we would have a weakening of the AMOC already sensible to our measurements. So we have a measurement in place right for that to measure the strength of the meridian overturning circulation. The one that is most used and has been more published is the one that from the rapid array at 26 north here close to Miami. And we have a component that is the Florida current measured by a telephone cable in the Florida Straits. And we have several sensors across the basin that measure them. But these measurements, they're very recent. They started this one in particular in 2004. And since then we built up several arrays, several transects across the basin that now we can measure this variability all across the basin and start to understand the relationship within the latitude. But they are still too recent to measure any sensible trend. So what we can do is to use historical observations, right? We have historical observations all the way from 1960 of temperature observations that we can be used to calculate the ocean circulation and heat content and sea level. And they're mostly in the upper ocean, the top 800 meters. And it has evolved over time, the technology to measure it. One of the technologies that is still in place is with the XBT transects that I work a lot with this project, that it has a few sections that can measure this variability of the meridian overturning circulation. And also we have starting 2004, 2005, these Argo floats. They're kind of little floats that goes from the surface to 2,000 meters and to the surface again. And we have now over 4,000 floats of these that measure temperature and salinity all the time in the ocean, every 10 days. In addition to the satellites, to the array of satellites that measure, we can have a view, a holistic view of the globe, what's being sensed at the surface. Now I will give a couple examples of some recent work that we have done. One is we published last year that use these Argo floats and these XBT, the expandable bathythermograph transects from Miami to Gibraltar Strait, and use the information together to try to estimate the strength of the meridian overturning circulation. And we see that using the information of both platforms, we can extend and construct a time series, a monthly time series all the way to 1995 most of the time. And then when we see that using both together, we strengthen the amount of information and capture better and a better relationship what's measured with the rapid array. And another recent work that was recently submitted by my colleague Shengfu Dong, and she used satellite observations, and from this observation use statistical relationships with temperature profiles, and from that we can estimate also this flux of the AMOC, not only in one latitude but across a whole bunch of latitudes in the Atlantic. And then we can extend this all the way to 93, where when these observations were available from satellite, and we can start understanding this latitudinal coherence and variability that from the AMOC. And finally, I will show some recent work I've been doing under the Climate Variability and Predictability program. This is a program that put together instrumental and paleo climate scientists, and we are trying to put together and the strength of the two kind of observations to understand the variability in the long term of the AMOC. And here, for example, we got several proxy data, which proxy data we can retrieve from sediment from corals or from shells, and in several different locations in the North Atlantic, annually resolved data. And with that, we can link that with the instrumental data, that the instrumental data from 1900, 1850 to the present, and then we can draw statistical relationships that we can actually link that to the AMOC variability, and not only extend that to the instrumental period, which is from 1850 to the present, but all the way to 1350, all the way to 700 years in the back, where we have this proxy data with the sediment core. And we see really that from our preliminary results that indeed the strength of the AMOC has been decreasing since the 1900s severely, and has not been seeing this rate of decrease for the past 700 years. So this is very interesting work, and I will stop here and thank you for the time. And thank you.
Thank you very much, Marlis. I remember learning about the thermohaline circulation my first year of grad school and finding it so fascinating. Thank you so much for that talk. We're going to move on to Estrella Malca. Estrella is a graduate of the University of Miami. She's a larval fish ecologist with CIMAS and the NOAA Southeast Fisheries Science Center's Larval Fish Lab. Her projects focus on the mostly secret life of larval fish, especially highly migratory species such as tuna, billfishes, and reef fish such as lionfish, groupers, and snappers. 2 years ago, Estrella joined the PhD program at NSU's Helmholtz College of Natural Sciences and Oceanography in Dr. Tracy Sutton's Oceanic Ecology Lab. She hopes to complete her degree by next year and continue learning about the earliest and most vulnerable life stages for all fishes. So Estrella, you can please share your screen and tell us a little bit about the life of larval fishes.
Thank you, Emily, for that introduction. I appreciate it. We're going to stay in the ocean for this presentation, but we're going to switch gears to live things. So I work with larval fish, and most of you have probably heard a fish, but you probably never thought about them being babies. So all fish that you've probably ever met and seen or eaten were babies at one point. So what I do is I basically use that as a tool and apply it to research and fisheries and management. So of course, our goal is to contribute to an integrated ecosystem-based approach using our larval fish as our main tool. However, it's quite tricky. There is a lot of fish in the sea, and we have a lot of different types and shapes, and they have different life stages. Some of them are found in the deepest ocean. Some of them float around in the surface water. So it's quite a challenge to study this field. A lot of these species are not described, so we understand some of the adult fishes, but we don't know what the babies look like. And we are learning new things, discovering new species daily. So it's a very exciting field. I encountered it as an undergrad at the University of Miami during my internships, and I started going to sea, and I fell in love with it. It's definitely my favorite thing is definitely to go to sea and participate and lead these surveys. These are some of the larval fish that we've collected in our surveys. And if you're interested in what they are, or some of the scary ones, feel free to contact me, and I'll be happy to, tell you what they are. You can tell me what's number 3 or number 23 or whatever. But I'll just bring your attention to the one that's circled. The orange is a lionfish. And in one of our surveys back in 2010, we actually collected the first larval lionfish. And some of you may be aware that lionfish are invasive in the Atlantic basin, and we unfortunately find them all the time now. So they're native to the Pacific, and they're believed to have been introduced through accidental aquarium releases over time, and now they're found. So this is an unexpected find, but they're beautiful fish. They have these beautiful fins, and they are very interesting. So we've recently had a paper published looking at their distribution in the invaded range. And feel free to contact me if you'd like to see a copy of that. It's available publicly. The challenge with larval fish is that there's so many different types and shapes and sizes. So taxonomy is the study of trying to figure out, how are they all related? What are their names? Are they in the same family, in the same species? What do they look like? Descriptions, photos now definitely are helpful. Genetics are tools that we use every day to discover more and more about them. So we have to learn a lot about each and every one of them, and then kind of put the puzzle together per se, and try to figure out how the ecosystem, the marine ecosystem as a whole functions. So some families of interest that our Larval Fish Lab has focused on is mainly in two realms. The reef fish realm. So these are fish that as adults associate with the reef, are bottom associated benthic fish, and other fish such as the mahi and the tunas that they live their whole life out in the open sea. So they have a pelagic lifetime. And just to pick one of the main and more exciting fish that we work with is the Atlantic bluefin tuna. And we've learned quite a lot about these guys over the last 15 years. We've been focusing on them. And we've learned about maternal condition. We've learned about their distribution, what is a favorable habitat for them versus unfavorable habitat. And we mainly use satellite data and ship-based data. And this is a picture of one of the NOAA ships that we utilize to do some of our research. And for the bluefin tuna, we've found that larval abundances over time are good indicators, and they help us figure out the strength of the population. So for example, in this map here, you have the Atlantic Ocean, and Atlantic tuna are strong swimmers. So they mainly feed in the North Atlantic Ocean, but when it's time to reproduce, they go a really, really long way just to go to the Gulf of Mexico or to the Mediterranean Sea. So they're definitely, putting a lot of effort to do this. So one of the main questions in our lab is like, why are they doing this? There must be a survival advantage for these larvae, for the adults to, use all this energy to do this. So we've been able to collect them and figure out a lot of different basic research questions, such as what are they eating? How are those different pieces in their stomachs? Believe it or not, one of our colleagues dissected their stomachs, very, very tiny work, all under hours and hours in a microscope. And they're able to figure out what are they eating, how much of it, are they successful feeders? And then also we've been able to use some of our larval tuna and answer some genetic questions. Can we figure out how related they are? So you can see how many adults potentially are spawning in the Gulf of Mexico using larval tissues. So the technology has certainly advanced quite a lot for you to be able to do this. And also our techniques to find these tuna have been improving over time. So we use these habitat maps and predictions, as well as in situ information to find bluefin tuna. But we don't only focus on these pelagic organisms. We also use, we focused on snappers and groupers and, as I mentioned, lionfish. So how do we collect them? First, we go to sea on the white ships, as I showed you, and then we use plankton nets as our main collection tool. We use different types of nets, but this is a bongo net. And this is a little video of what it looks like to be in the net. So if you were a little baby larval fish, maybe you get caught in that, and then, we'll collect you. And you do this hundreds of times. You tow the net, you rinse, repeat. This is a typical sample of a plankton jar. And this is more or less what you would see. Right here we have a little baby barracuda in that little circle that you see. So this to me indicates that it was likely a coastal sample because they're getting ready to come closer to shore and perhaps mangrove or some seagrass habitat. All of the other stuff that you see is all either copepods or other invertebrates. And for example, on the right here, we've got some baby tuna and that we've picked out and put them aside so that we can do some of our experiments with them. But all of that is floating around in the ocean just outside Erasmus. One of the big projects that our lab is currently working with is the Bluefin's Gulf of Mexico project. So this is a NOAA Restore Science Program funded project with many different universities, University of Miami, CIMAS being one of them, of course. And my piece is focuses on feeding and growth, but there are many different pieces to this particular project, and mainly looking at the biogeochemistry, nutrient sources, the phytoplankton composition, zooplankton abundance, and looking a lot at stable isotopes as a tool to figure out the food web dynamics. And basically, we're using bluefin tuna as the top of the food chain and trying to figure out all the different links and model the results. So what do I focus on? I mentioned I work on otoliths. So what are otoliths? They are these little tiny stones, and you can see it. They're kind of shining in that little video. So all bony fish have otoliths, and as larvae, you can examine them under a microscope. And at least for bluefin, I can actually count how many rings they are. So I spend a lot of time in the microscope trying to prepare and age these fish. And another aspect of our lab work in both in the field as well is that we support and mentor a lot of undergraduate and graduate research. And over the years that I've been involved with the lab, as a student, as a scientist, we've mentored multiple dozens of students, brought them to sea, taught them many different techniques, had them basically do everything, run the heavy, expensive equipment, as well as, do the heavy lifting, load the ship, unload the ship, sort the sample, learn how to ID, learn how to pull otoliths. And most of the students here, some of the most recent ones are either gone on to grad school or have jobs now. And we definitely miss going to sea, and we hope that, at least I hope I can return soon. But maybe next year. So, but with that, thank you for your attention. If you have any questions about our research, feel free to contact me. And I have a little screenshot there of our blog at the bottom that we talk about some of our adventures and the different projects that we've had. Thank you.
Thank you very much, Estrella. That was really interesting. And also thank you in particular for highlighting some of the student research that goes on as an incredibly important component. Our next speaker is Nikki Vollmer. Nikki received her PhD from the University of Louisiana at Lafayette. She's currently a CIMAS scientist at NOAA's Marine Mammal Molecular Genetics Lab in Lafayette, Louisiana. She has been studying marine mammals for over 20 years in places like Shark Bay, Australia, the Gulf of St. Lawrence in Canada, and the Gulf of Alaska. Most of her research is focused on studying populations of dolphins and whales using DNA and answering questions that are important for their conservation. So thank you very much. Take it away, Nikki.
Thank you, Emily. All right, so today I'm going to talk about the conservation genetics of bottlenose dolphins in the Gulf of Mexico. But first off, I want to introduce you to the bottlenose dolphin, which is also known by the Latin name Tursiops truncatus. So they're found in temperate and tropical waters all around the globe, including off of both coasts of the US and in the Gulf of Mexico. And in this map here of the Gulf of Mexico, they occur pretty much anywhere you see that light blue color. So close to shore, in bays and estuaries, but also out onto the continental shelf and slope. So bottlenose dolphins, like many marine mammals, they have the capability to disperse pretty much anywhere they want within their environment. So this means the dolphin swimming off of Texas there's no reason why it can't swim across the Gulf of Mexico all the way to Florida. But some of the questions I try to answer with my research include, do these animals go anywhere they want just because they can? Or do they interbreed with other dolphins from faraway places? In other words, is the same population of bottlenose dolphins that lives off of Texas also found living in the waters of the Florida Keys? So why are questions like these important for conservation? Well, because bottlenose dolphins inhabit such a wide geographic range, they're exposed to many different types of threats which can negatively impact their populations. And you can see I've listed some of the big threats here, and you can see them in some of the pictures as well. But because oftentimes these threats are not evenly distributed across the entire habitat that a dolphin uses, it's important to identify the different populations, figure out what threats they overlap with, and how they're being impacted by them. And this type of information is necessary to develop conservation and restoration strategies. So you might ask, how does one study dolphin populations? And the answer is by looking at their DNA. And we can get DNA from dolphins in lots of different ways. For example, we can extract DNA from the skin collected from dead stranded animals on the beach. We can also extract DNA from bones and teeth of museum specimens. But most often we use biopsy darts to get small tissue samples from wild animals. And we extract the DNA from the little bit of skin that we get from these biopsies. And you can see what a biopsy looks like in this bottom picture. So now I want to tell you about a recent project where I used DNA to look at bottlenose dolphin populations in the northern Gulf of Mexico and how they were impacted by the Deepwater Horizon oil spill. But first, you need to know that in the United States, we utilize the concept of stocks to help manage marine mammals. So a stock is a group of the same species in a common area that interbreeds when mature. Now, often these stocks are initially set up primarily for management purposes. Ideally, they would represent the real biological populations that are out there, but when they're first set up, they don't have to. And currently within the Gulf of Mexico, there are 36 stocks of bottlenose dolphins that have been delineated, and you can see those 36 stocks in the two pictures below. There are 31 stocks in bays, sounds, and estuarine areas, and those are the little black squares you see on the left. And then there are 5 coastal and offshore stocks, which are the colored areas on the right. Now, these stocks were mostly set up based on differences in geographic and oceanographic features. And for many of them, we haven't yet been able to incorporate information about the biological populations because we just don't know that much about bottlenose dolphin population structure in the Gulf of Mexico. All right, of those 36 stocks, there are 3 that I want to focus on here in the northern Gulf of Mexico. And this is an area where we know the oil slick overlapped a lot with where dolphins occur. There's the Mississippi Sound Lake-borne Bay Boudreaux stock, which is in red, and I'm gonna just abbreviate this as the Mississippi Sound stock moving forward. There's the northern coastal stock in purple and the continental shelf stock in yellow. Now, right after the oil spill, calculations using the abundance estimates from these stock delineations calculated that about 22% of the dolphins in the Mississippi Sound stock, 38% in the northern coastal stock, and 4% of the continental shelf stock were killed due to the Deepwater Horizon oil spill. But at the time, it was unknown whether or not these stocks were truly representative of the real populations that are in these waters. So I looked at all of the samples that we had from these stocks, from these 3 stocks in the Northern Gulf, and specifically I looked at 2 types of DNA from all the samples. I looked at nuclear DNA, which is inherited from both parents, and I also looked at the mitochondrial DNA, which is only inherited from the mom. And when you look at both types of DNA in a group of individuals, you're able to tell which ones are closely related to one another. And thus, you can identify genetically distinct populations. So I looked at the DNA for 583 samples, and after running a whole bunch of statistical tests, determined that the best number of populations given these data was just 2. So here I color-coded all of the samples based on the population they were assigned to. And I think you can see that there really is no exclusive Mississippi Sound stock or Mississippi Sound population, nor is there a population that only occurs within the boundaries of either the northern coastal or the continental shelf stocks. But what the genetic data did find was a population, which I just colored green, that's found mostly west of Mobile Bay, Alabama. It's found in the Mississippi Sound, but also in adjacent coastal waters. And there are even some individuals from the green population found along the Florida Panhandle and around St. Joseph Bay, Florida. And then we have the blue population, which is found mostly east of Mobile Bay, and on average is found in deeper waters, typically greater than 20 meters depth. So finally, I wanted to reinterpret impacts or rethink about the impacts from the Deepwater Horizon oil spill, given the ranges of these populations, rather than strictly thinking about the current stock delineations. So what you see here on this map is the cumulative surface oil detected in the months following the Deepwater Horizon explosion. And again, I'm showing you those mortality estimates for the Mississippi Sound and northern coastal stock. But you might think of these estimates a little differently if you consider the actual ranges of the populations that occur in these stock areas. For instance, with the green population, you can see that a large portion of it, which includes the Mississippi Sound, overlaps with the oil footprint. So this suggests that more individuals from this population were exposed to oil than previously thought. In fact, if you consider all of the individuals in the green population that you see here, compared to just those within the boundary of the Mississippi Sound stock, the green population was exposed to over 3 times more oil on average. And a similar situation also occurs with the blue population if you compare it to just the individuals within the boundaries of the northern coastal stock. Therefore, it's possible that the overall health impacts on bottlenose dolphins from the oil spill in this part of the Gulf of Mexico were actually underestimated given the real range extent of these populations. All right, to conclude, genetic data support the presence of 2 populations of bottlenose dolphins within waters of the Mississippi Sound and northern Gulf of Mexico. However, neither of these populations align with current stock delineations for the Mississippi Sound, northern coastal, or continental shelf stocks. So this suggests that some revision is necessary so that we are accurately managing the real populations of bottlenose dolphins in these waters. And finally, this is a great example of how knowledge of population structure really is critical for understanding impacts in the marine environment, and is really important for effectively planning future restoration and conservation strategies. Thanks, and I'm happy to try to answer any questions you guys might have.
Fantastic. Thank you so much, Nikki. And thank you for teaching me Tersiops truncata. I'm going to trot that out a lot. It's a great expression there. I am going to thank all of our speakers. That was really even better than I'd hoped. So thank you all very, very much. And I will pass the mic back to Dean Avasar, who has some closing remarks for us. Thanks very much.
Thank you so much, Emily. Thank you, everybody, for attending this very interesting evening, and for all the speakers, of course, and to Director Ben Kurtman for the presentations. I think it was fascinating and very interesting. I have seen a few questions being asked, and most of them are being answered in writing. We will give maybe times for 1 or 2 final remarks through questions that will be selected from our Director of Communications right after this. What I would do right now is encourage everybody that is interested in this series to come to our March 9 presentation. It will be addressed by Professor Catherine Mack. And the title of her presentation is Climate Change 2021, Managing Floods, Heat, and Fires to Keep People and Nature Safe. Thank you, everybody, for participating. And I believe that we will have only time for 1 or 2 questions very quickly. So, Diana, please go ahead.
Thank you, Dean Avasar. Thank you all for attending. So yeah, there's a lot of questions here. So real quickly, we'll get with the CIMAS folks. Those we don't answer or have a chance to answer in writing in person, I see many of them are being answered. We'll get back to you in writing. There's a question from Matthew for Nikki. Are dolphins territorial? Do they tend to stay in the same area? Thank you.
Oh, that's a great question. As far as Tursiops truncatus bottlenose dolphins, I'm not sure we really know that for sure. There are some other types of bottlenose dolphins that live in Australia, and they are known to form male bonds or male alliances. And they can be a bit territorial, but mostly the territory they're protecting is females. And who they're going to mate with, not necessarily the area or the environment they're trying to protect.
Nikki, are you going to look at the Louisiana population and by chance try to work with Louisiana Coastal Wildlife Network at Audubon for dolphin necropsy samples?
Another good question. Yeah, so Louisiana population, we're not 100% sure there is an exclusive Louisiana population, but one of the projects we're working on right now in our lab is specifically focused on dolphins in Barataria Bay, Louisiana. So yeah, we are reaching out to whoever we can locally and across the Gulf to help get samples and coordinate with.
Quick question for Marlos. Can Marlos explain in more details the dipole gyres of warm and cold waters off Greenland and Newfoundland in relation to the Gulf Stream moving further south? Do these gyres exist normally?
Thank you. I was exactly answered here on the chat. Yeah, this gyre, the gyre of Greenland is known as the subpolar gyre, and we have this exactly this migration of the Gulf Stream that is related to that variability and the amount of heat that is crossed to the subpolar gyre.
Excellent. So we will include all of these questions and answers in the follow-up email that we'll send to you. We'll print out or basically extract the text and the questions from the Q&A. Last but not least, there's been a lot of requests for the ability to communicate with the presenters tonight. So if all the presenters at this time could just take a moment and put your email addresses or the best place for folks to continue to ask you questions, that would be appreciated. And I think with that, we are only 3 minutes over schedule, but we will see you on March 9th. Dr. Catherine Mock's presentation, and as a registrant and an attendee of these Sea Secrets meetings, we always do a follow-up email and we include the link to be able to review and watch the video of these presentations that will live on our YouTube channel. With that, we are adjourned. Thank you so much for joining us, and we'll see you on March 9th.